Dear students, welcome to all of you. Today we are here to discuss biological membranes. First, the introduction. The word membrane comes from Latin word membrana that means a skin. Today's word membrane has been extended to describe a thin flexible sheet or film acting as a selective boundary between two phases because of its semi-permeable properties. The membranes are active structures surrounding cells and most eukaryotic organelles such as endoplasmic reticulum, the Golgi apparatus, vacuoles, mitochondria, chloroplasts, and the nucleus. Regardless of their nature, prokaryotic or eukaryotic, the membranes are integral to the structure and function of all cells. The membranes define the external boundaries of cells and regulate the molecular traffic across that boundary. In eukaryotic cells, they divide the internal space into discrete compartments to segregate processes and components. They organize complex reaction sequences and are central to both biological energy conservation and cell-to-cell communication. The membranes are flexible, self-sealing, and selectively permeable to polar solutes. With their ability to break and reseal, two membranes can fuse as in exocytosis, or a single membrane enclosed compartment can undergo fission to yield two sealed compartments as in endocytosis or cell division without creating gross leaks through cellular surfaces. Because membranes are selectively permeable, they retain certain compounds and ions within cells and within specific cellular compartments while excluding others. Now, the history of biological membranes. The term membrane as an invisible film that surrounds a cell and serves as a barrier between cell contents and the environment was first used independently by botanists Van Mohl and Van Nageli in 1855 for explanation of plasmolytic phenomena. Between 1895 and 1902, Overton measured cell membrane permeability for many compounds and showed a direct relationship between the ability of these compounds to penetrate through membranes and their solubility in lipids. Gotter and Grindel in 1925 showed that the area of the monolayer of lipids extracted from erythrocyte membranes is 2 times larger than the total area of erythrocytes. Gotter and Grindel extracted lipids from hemolyzed erythrocytes with acetone. They evaporated the solution on the surface of water and measured the area of the formed monomolecular lipid film. The results of these investigations suggested that lipids in membrane are arranged as a bimolecular layer. This supposition was verified by investigations of the electrical parameters of biomembranes like high electrical resistance and high electrical capacitance. At the same time, there were experimental data that testified to the fact that biological membranes contained protein molecules as part of their composition. These contradictions in experimental results were removed by Donnelly and Davison in 1935, who proposed the so-called sandwich or bread and butter model of membrane composition. Donnelly and Davison postulated the presence of a protein layer attached to the polar head groups on either side of Gotter and Grindel's bimolecular layer of lipid sheet. The presence of protein contributes to the stability and strength of the lipid film comprising the membrane. Dear students, now we will discuss the structure of biological membranes. Several models have been proposed to explain the arrangement and the structure of lipid and protein molecules in the plasma membrane. On the basis of its lipid permeability character, Overton in 1902 regarded it as a lipoidal membrane. Gotter and Grindel in 1925 regarded it as a lipid bimolecular layer. Thereafter, several important models were proposed to explain the physical and biological features of cell membranes. For example, butter sandwich model of Denley and Davison, unit membrane model of Robertson, micellar or mosaic theory of Hilaire and Hofmann, protein crystal model of David Green and his co-workers, lipid globular protein mosaic theory of Roderick and Capaldi, and fluid mosaic model of Singer and Nicholson. The fluid mosaic model of plasma membrane is most widely accepted. Dear students, because of the wide acceptance of fluid mosaic model, we will discuss here it in detail. The fluid mosaic model of biological membranes was proposed by S.J. Singer and G. Nicholson in 1972. According to this model, a plasma membrane is a continuous lipid layer having integral protein molecules. The two, that is, lipids and proteins, are so dispersed that they form a mosaic pattern. The biological membranes are semi-fluid structures where lipid and protein molecules perform translational movements. The butter sandwich model of Denley-Davison assumes hydrophilic binding between protein and lipid molecules, whereas fluid mosaic model considers this to be hydrophobic. The membrane has globular proteins which may be intrinsic or extrinsic. Many intrinsic proteins and phospholipids of the membrane show amphipathy, that is, they have both hydrophilic and hydrophobic groups within the same molecule. The nuclear magnetic resonance (NMR) and electron spin resistance (ESR) studies show that the membrane is dynamic. The lipid tails show flexibility. The molecules can rotate or show flip-flop motion. Singer and Nicholson proposed that the proteins that are integral to the membrane are a heterogeneous set of globular molecules, each arranged in an amphipathic structure, that is, with the ionic and highly polar groups protruding from the membrane into the aqueous phase, and the non-polar groups largely buried in the hydrophobic interior of the membrane. These globular molecules are partially embedded in a matrix of phospholipid. The bulk of the phospholipid is organized as a discontinuous fluid layer, although a small fraction of the lipid may interact specifically with the membrane proteins. The fluid mosaic structure is therefore formally analogous to a two-dimensional oriented solution of integral proteins or lipoproteins in the viscous phospholipid bilayer solvent. In the fluid mosaic model, a mosaic of proteins float in the fluid lipid bilayer like boats on a pond or icebergs in sea. Dear students, now we will discuss the composition of biological membranes. Depending on the cell type, function, and species, the thickness of biological membranes will vary from approximately 2 to 10 nanometers. Regardless of their source, all biological membranes have a number of structural characteristics in common. The main feature of all membranes is their phospholipid bilayer organization. The biological membrane contains about 20% to 70% lipids whose constituents are mainly phospholipids, cholesterol, and glycolipids. Their proportion varies in different cell membranes. The phospholipids, which make up 55% and 75% of the total lipid content, consists chiefly of lecithin and cephalin. The remainder consists of sphingolipids and glycolipids. Phospholipids are unique molecules in that one end of the molecule is polar or hydrophilic, while the other end being nonpolar or hydrophobic. Proteins are integral components in the composition of membranes. For example, 76% of the inner mitochondrial membrane is protein and 24% is phospholipid. The plasma membrane from human red blood cell contains 44% proteins and 43% phospholipids. The myelin from neurofiber contains about 18% proteins with 76% being phospholipids. These membrane proteins participate in a number of fundamental roles such as receptors, enzymes, and as transport proteins. The two basic types of membrane proteins are integral or intrinsic proteins and peripheral or extrinsic proteins. The integral proteins are those proteins which are tightly bound to the membrane. They represent more than 70% of the two protein types and penetrate the lipid layer wholly or partially. The integral proteins may be attached to the oligosaccharides to form glycoproteins or to phospholipids to form lipoproteins or proteolipids. Common intrinsic proteins are rhodopsin found in retinal rod cells and cytochrome oxidase found in mitochondrial membranes. On the other hand, the peripheral proteins are loosely bound to only one side of the lipid bilayer. They are also called extrinsic proteins and can be separated by addition of salts. They are soluble in aqueous solutions and are usually free of lipids. Common examples of extrinsic proteins are cytochrome C, found in mitochondria, acetylcholine esterase, found in electroplaques membranes, and spectrin, found in erythrocytes. About 30 enzymes have been isolated from various plasma membranes. Those most constantly found are thionucleotidase, sodium-potassium activated ATPase, alkaline phosphatase, adenyl cyclase, RNase, and acid phosphomonoesterase. The membranes of eukaryotic cells usually contain between 2% and 10% carbohydrates in the form of glycolipids and glycoproteins. About 5% carbohydrates have been found in the plasma membrane of liver cells and RBCs. Hexose, hexoamine, fucose, and sialic acid are the commonest carbohydrates found in the membrane. Now we will discuss the biological membranes and the transport of molecules. One of the integral roles attributed to membranes is the regulation of materials into and out of the cell or membrane-bound organelles. Due to the amphiphilic nature of the phospholipids, the ability to traverse a membrane is a function of both the size and polarity of the molecule. Whereas very small non-polar molecules are able to diffuse rapidly across membranes, polar molecules experience a certain degree of difficulty. The uncharged polar molecules less than 90 daltons in size are capable of diffusing across membranes, whereas those greater than 90 daltons in size do not. Ions, however, are incapable of diffusing across the membrane regardless of their size. From this, it is apparent that the movement of substances across biological membranes involves a number of different mechanisms. The major cellular transport mechanisms are passive transport and active transport. First, the passive transport. The process whereby substances move from one side of a membrane to the other without the expenditure of energy is referred to as passive transport or diffusion. In this type of transport, the movement of an uncharged substance across the membrane is based solely on differences in concentration. This concentration gradient determines the direction of transport, which is from an area of greater concentration to one of lesser concentration. In addition to the phospholipids, proteins are important to the structural composition of membranes. Not only are there a number of different functions attributed to membrane proteins, but there are also a number of different ways in which these proteins are associated with the membrane. Transmembrane proteins, whether single pass or multiple pass, span the membrane with portions of the protein on either side of the membrane. It is believed that one role of these transmembrane proteins is to mediate the transport of substances across the membrane. They can act either as carrier proteins or as channel proteins. A carrier protein functions by binding to a specific uncharged substance and then by undergoing a number of structural changes and is able to transport that molecule across the membrane. Channel proteins form a channel through which polar molecules or ions can pass from one side of the membrane to the other. A discrete set of channel proteins, the ion channel proteins, form channels or pores through which ions of a specific size and charge can pass. To regulate the flow of ions through the ion channel proteins, these transmembrane proteins have the ability to open or close the pore through which the ion is transported. These are referred to as voltage-gated channels. The difference in the concentration of ions on either side of a membrane creates an electrochemical gradient or membrane potential. In general, the interior of a cell tends to have an overall negative charge with respect to its exterior environment. This potential difference favors the transport of positively charged substance into the cell. Therefore, the transport of charged molecules or ions across a membrane is not only based on concentration difference, but it is also based on its membrane potential. Now, the active transport. The transport of substances against a concentration gradient is known as active transport and requires both a carrier protein and an energy source. These transmembrane carrier proteins have been classified as uniport, symport, and antiport carrier proteins. The uniport carriers transport a single molecule from one side of the membrane to the other. Both symport and antiport carrier proteins involve the dependent transfer of two molecules, and as such, they are also referred to as co-transporters. Symport carriers transport two molecules in the same direction, such as amino acids and sodium. Antiport carriers, in comparison, transport the two molecules in opposite direction, as in the case of sodium and potassium. The transport of macromolecules such as proteins and polysaccharides rely on vesicular transport. In this type of transport, membranous vesicles are formed around the substance or substances that are to be transported into or out of the cell or membrane-bound organelles. Where endocytosis refers to vesicular transport into the cell, exocytosis is the vesicular transport of substance out of the cell. The energy required for the active transport of molecules across a membrane can come from the hydrolysis of adenosine triphosphate or from specific ion gradients. Active transport can be classified into primary or secondary active transport. The energy released from the hydrolysis of ATP drives the primary active transport mechanism, whereas ion gradients drive the secondary active transport mechanisms. There are several different classes of primary active transport, which include P-class ion pumps, F-class ion pumps, V-class ion pumps, and the ATP-binding cassette or ABC superfamily. The ABC superfamily is responsible for the transport of a variety of substances. Secondary active transport may include either symport or antiport co-transporters and are responsible for the transport of such substances as amino acids and sugars. Dear students, now we will discuss the functions of biological membranes. In all living cells, biological membranes carry out the functions of barrier that divides the cell from the environment and the internal cell volume into isolated compartments. The barrier properties of membranes and working of membrane pumps cause a regular distribution of ions between the cell and extracellular medium, which lies in the basis of the processes of intracellular regulation and signal transfer in the form of electrical impulse between cells. A common feature for all membranes is the function of mounting plate or matrix on which there are proteins and protein groups that are disposed in a definite order and create electron transfer, energy accumulation in the form of ATP, regulation of intracellular processes by hormones coming in from outside and intracellular mediations, recognizing of other cells and foreign proteins, light reception, mechanical effects, etc. A flexible and elastic film which lay the basis of all membranes also play a definite mechanical function, keeping the cell intact under mild mechanical loads and disturbances in upsets of osmotic balance between the cell and environment. Common for all membranes, functions are barrier for ions and molecules, and matrices for protein groups are mainly provided by the lipid bilayer that has in principle the same structure in all membranes. Nevertheless, the set of proteins is unique for each membrane type, which allows membranes to take part in carrying out various functions in different cells and cell structures. Dear students, these were the general functions of biological membranes. Now we will switch on to functions of biomembranes which are used in food technology. First, biomembranes and rehydration. Dry phospholipid bilayers are known to undergo transient, that is phase transitions and permeability changes in dry membrane during rehydration. Number second, liposomes. Liposomes, spherical bilayer vesicles, is a good example of biomembrane functioning in food industry. Liposomes have been used to deliver food flavor, nutrients, and more recently, food antimicrobials that could aid in the protection of food products against growth of spoilage and pathogenic microorganisms. Number third, unique interaction of trehalose and biomembranes. Trehalose, a non-reducing diglucose sugar found in nature, confers to certain plant and animal cells the ability to survive dehydration for decades and to restore activity soon after rehydration. The interaction between trehalose and cell membranes or proteins is subject of recent research to elucidate the mechanisms in this unique behavior of preservation. Number 4. Biomembranes and pulsed electric field processing, PEF. The ever-increasing trend toward nutritionally qualified food has challenged food technology to produce fresh-like foods by replacing thermal treatments. PEF is used to kill or inactivate microorganisms based on the principle of membrane fluidity and disruption. PEF is successfully used in processing and facilitating juice extraction from fruits, also based on structural properties of biomembranes. Number 5. Biomembranes and chill injury. Membrane-associated disorders of plants include chilling, freezing, and desiccation injuries. Mitochondria and other cell organelles of plant cells exposed to low temperature and other biotic and abiotic stresses produce superoxide hydrogen peroxide in the cells. This superoxide hydrogen peroxide can diffuse throughout cell, causing peroxidation of membrane lipids, which results in membrane disruption, increased permeability and metabolic disturbances, and eventually the visible symptoms of chilling injury. Chilling stress is responsible for oxidative stress, during which the reactive oxygen species ROS are generated and affect the biomolecules and the plasma membrane. Scientists have demonstrated that oxidative stress is an early response of cucumber fruits to chilling and that lipid peroxidation may contribute to the development of chilling injury. The polyunsaturated fatty acids, linoleic acid and linolenic acid, are particularly susceptible to attack to ROS, giving rise to complex mixtures of lipid hydroperoxides. The peroxidation of lipids is considered as the most damaging process known to occur in living organisms. Dear students, this is all about biological membranes. We have discussed over here history, composition, and structure of biological membranes. We have also discussed the functions of biological membranes, especially with regard to food technology. Thank you very much. God bless you.